So I want to talk today about innovation in the grain industry, and in particular some innovative products and processes and projects. So today I'll talk about 4 different projects or programs, and all the way through I tend to use this little line here to talk about how the pipeline of products move from a proof of concept through an early development, advanced development, integration stage, commercialisation itself, and then looking at uptake and delivery. And then the project itself, the contracted project with the researcher, there's a little bar here giving you the years, the financial years that it's actually funded for. And in the case of some projects, not much is achieved by the looks of it in a 4-year, and then you might have another 4-year after it, or you might be running 2 or 3 different projects off the back of the original platform technology to actually deliver more than one product. So first of all, I want to talk about insecticides. Now the comment I want to make here, looking through this long list of key groups of insecticides, was resistance. DDT was brought out in the 1940s, and by 1948, 6 years after its release, we had the first resistant insects appearing to DDT. All the way through, we've lost these products in key insect groups, all the way through to the latest generation of products, the neonicotinoids like Confidor. We have had significant resistance appearing in certain insect groups. So we've been fighting a losing battle in many respects in some of these areas. The result being is we have lots of gaps or potential gaps forming in specific areas. Now some of the ones that you guys will be very aware of in particular are things like pulses, where not a lot of products actually get registered. And when we lose a product in the pulse area, it's a big gap left behind. The products coming through, well, there's not a lot in the pipeline. You talk to the chemical companies out here this afternoon and they'll tell you there's not a huge lineup of insecticide products coming through that pipeline at the moment, which is a little bit frightening. And the main reason being is it's going to cost about $200 million to bring a new active to market. That's what the big guys tell me in the last couple of years. And most of that money has actually been spent on the toxicology, residues, all this sort of area. That's the expensive part. It's about 10 years, well, in some cases, trying to get it through our registration process, you might as well say 15. And the chances are about 1 in every 140,000 molecules out of a screening process actually gets through and makes it as a product. So it's a pretty damn tight screening process, starting with a big number and ending up with a very small number. Again, expensive. So this is what a pesticide pipeline looks like to a chemical company. There's a lot going on in different areas, and as you can see in the last couple of years, this is the area that's really sucking up the money. All this toxicology work down the bottom here. So the one I want to talk about today is one of the projects that we started a couple of years ago, back in 2010. It was a 4-year project at this stage, working with the Institute of Molecular Biology at Queensland University. And the idea on this project was to develop a new class of insecticidal compounds based on short-chain peptides found in spider toxins. So we had to invent, first of all, a whole set of new bioassays to screen toxins and look for the elements they're after, isolating those peptides and then developing the products. So why spiders? Spiders are the master insect killers. They've been around for a long time, and they are the best at killing insects. They've got an enormous range of toxins. If we look at, they've been around for 350, 400 million years. There are a lot of species, and each individual spider has an individual venom, and each of those venoms can have up to 500 different peptide compounds which are insecticidal. So they have a huge range of insecticides that they're using, and they've developed these to ensure that there is nothing out there that's resistant to a spider bite. And they're very good at doing it. So the potential for us is anything up to 10 million different insecticidal peptides out there that we could be looking at. So open that screening door up again and let's let some new ones in. Some of those peptides and some of the ones that we've looked at so far actually fit across a wide range of groups. So things like a sodium channel blocker. Now sodium channel blockers are known to you guys already. The synthetic pyrethroids are sodium channel blockers, and spiders likewise have these particular toxins which are known to be sodium channel blockers. But what's really interesting is they have potassium channel and hybrid toxin channels and calcium channel blockers. There are no insecticides on the market that are a calcium channel blocker. We'd love to have one. And why do we like these particular peptides? Well, they have a cysteine knot in them, sort of represented by this sort of figure here, and cysteine knots make them very robust. They're able to resist proteases, and that means when they're injected into an insect, the insect can't metabolize them away easily or quickly. They can withstand temperatures. We can boil these, and it won't actually break them down and denature them quickly. And they're tolerant of high pHs and harsh solvents. Fantastic. This means we could probably tank mix and do things like that down the track as well. So we started off with a large collection of spiders from all around the world. We have a few scorpions in there as well, just to mix it up a little. These spiders are mostly what they call the ancient line of spiders, so these are not the web-forming type. These are more likely to be tarantulas, trapdoors, funnel webs, those types of spiders that rely much more on their venom to kill an insect than they do wrapping it up in silk. So using that group of spiders and some of the skills of the team, where are we up to now? We've chosen the pests that we want to chase, and we've given them a short list of what are the key insect pests for our industry. We've gone through and we've selected out a short list of peptides and isolated those, and we're running it through a very high throughput screening mechanism, and this is part of the real success of this program, was to develop that high throughput screening mechanism based on insect stem cell lines. And we're at this point now of trying to decide which way we're going to go. There's a potential to produce a transgenic yeast which will actually produce the peptide. You spin the peptide out of that mixture and spray it on as a product in that format, or potentially we could also put that gene into the plant itself and have plants expressing it. Now both of these are still expensive pathways, but this one's probably a much more expensive pathway. But potential for things like sucking insects is fairly high on that one. So coming along, but still very early days. The next area I want to talk about is infrared. Now, infrared is a standard platform for you guys, and many of you, if you don't know the Infratech, you possibly know the Green Seeker, all based on infrared, near infrared or mid-infrared. Both have become widely accepted in agriculture. The biggest issue of using infrared in agriculture is the cost. These machines are not cheap. The size of them, the optical units inside them are large and clunky. The robustness, they just don't handle being knocked around very easily because they're based on optics. And the speed. In the case of some of these machines, they're taking anything up to a minute to take a single reading, which is far too slow for where we want to be. If we really want to unlock the value out of IR, we need to move away from what this clunky optics inside these machines is and go to something a little bit smarter than that. So our challenge was to develop a new IR detector in this project. So it needed to be fast. It needed to be able to measure what we're looking at in microseconds or even nanoseconds as opposed to minutes. It needed to be able to be robust. It needed to be cheaper. The inside optics in some of those more expensive machines is around $10,000 or more. We wanted to get the actual optics, that componentry, down under $100 to get it into small, cheap handheld instruments. And small. Can we get the whole detector unit under 1 centimetre in size? So we've been going on this project since 2009. And at the moment, we've progressed to the point at this stage where we have a prototype at the moment. This is just a schematic look at it. When you look at it under the microscope, it's only 200 micron across. And there's one inside here. It's come along a long way. And this is what it looks like as a chip. Well, that's the actual chip inside there. And this is the detector itself. So how does it work? You've got the light usually from a halogen lamp or an LED, either reflecting off a surface or being transmitted through a surface and hitting these mirrors. These are 5-layered mirrors, and it's all about the light bouncing between the two mirrors. By moving the mirrors closer or further apart using voltage, we can actually then start to look at a spectrum. So as the mirror moves, it's tuning the wavelengths, and this gives you the wavelengths of the spectrum of whatever you're looking at, and that's how IR is measured. So where are we going to go with this? What are we going to do with this little widget? So if we can build something that just has a simple light source, a range of spectrometers, these, and some capacity to actually convert that into a digital image and some optical fibres. I think we can develop a whole range of handheld, cheap units based on IR. So one of the things I've said to the group that we wanted to look at was dumb probes. So let's not try and reinvent the whole idea of having to have a handheld instrument and develop all the battery technology, develop all the processing capacity. We've got that in a thing like an iPhone. Why don't we just plug it into that? There's the source of electricity, there's the processing power, and guess what? It'll give me a GPS reading to boot. Now, what do we want to measure? Well, the ability to have these units inside your silos and be able to measure phosphine. We know that IR measures phosphine quite effectively, so we could have these spread through the whole silo and tell you where your phosphine front is. Could we have it testing for moisture and protein inside the silo as well, so you know what the grain is going in the top and you know what the load looks like. But the number one that we're starting on is a soil probe. Now, the challenge I've sent to the engineering team at the University of South Australia is I want a probe that'll go to a minimum of 60 centimetres, and it'll have a minimum of 10 of these, or 6 to 10 of these microspectrometers up and down that probe. So it measures soil moisture, nitrogen and carbon, which are all three elements very easy to be measured by IR at 10 centimetre bands down the soil. Now, they've come back and they're saying that's very possible, and we're starting to look at building prototypes. I'm also looking at something that we can use to measure nitrogen in leaf as well. And the idea would be both of these probes are cheap and able to be plugged into your iPhone and send that information off to perhaps you guys to give it interpretive data to send back to the farmer with the georeferences on it. The next project I want to talk about is about novel endophytes. So these are unusual endophytes, an endophyte being a fungus that lives inside a plant. These are unusual in that they never leave the plant. They live their entire lifespan inside a plant. In certain plants, these are quite common. So ryegrasses and fescues often have endophytes in them, and in fact it's an endophyte that causes ryegrass staggers. What do they do for the plant? Well, it's a symbiotic relationship. They often provide hosts protection against abiotic and biotic stresses. In particular, insects, nematodes and plant pathogens, they have some strong effect on those. And they can also increase plant productivity and persistence by improving the root system. They have all sorts of abilities. These have been in the plant world for millions of years, but it's only something that we've discovered in the last 15 years and started to make use of. So the project really started almost 15 years ago with a group in New Zealand when they were looking at ryegrass staggers and the alkaloids that are produced by these particular endophytes were causing this problem in sheep. So the first thing they did was, why not take the endophyte out? Unfortunately, when they took the endophyte out, they discovered the plants didn't grow particularly well. So it was a required symbiotic relationship for that plant to be successful. So with the endophyte, they had a problem. When they had no endophyte, the sheep were okay, but we couldn't get the pasture to establish. So the next step was to start to look for very specialised endophytes that no longer produced the nasty alkaloids, the ergovalines and the lolatrims, and produced other alkaloids that had a similar effect in terms of they allowed the plant to establish and control insects, but didn't have a problem with the sheep. And that's what they successfully did. The chemistry, and it is all about chemistry in this regard, is unique to some of these endophytes. They produce a whole range of bioactives. They're mostly alkaloids. The type of alkaloids depends on the fungal strain present, and the host plant also has a relationship in terms of how the alkaloids are produced and how much is produced. The chemistry is diverse. We've only really just touching the beginnings of where we could go with this. We know there are lots of different compounds that have been produced, many of which you don't yet understand. So some of the ones that are actually on the market in ryegrass are AR1, which was probably released more than 10 years ago now, and it controls Argentine stem weevil and mealybug. The latest one they put out, AR37, and some of the strains that have come out of meadow fescue control an enormous number of different insects. So where do we go for our project? Well, I want to put these types of endophytes back into cereals. When we say back into cereals, we know that they were in cereals thousands of years ago because some of the precursors to our modern cereals still have endophytes in them. So the skill sets from this team are, and it is quite a large team in New Zealand running this project, cover an enormous range of different areas. There's a lot to be done in this project. Where are we up to? Well, at this stage, we've gone past the discovery. We've gone and collected a whole heap of samples from Georgia and Armenia and Turkmenistan. These are ancient cereal lines or close relatives of cereals, and we've searched through and found a whole heap of successions or accessions with endophytes in them. We've gone through and described and looked at the diversity of those. We have, 18% totally unique genomes in those endophytes. These are things that have never been seen before. And the chemistry, we have a lot of lolines, and lolines is one that we're chasing because we know it's a powerful anti-insecticide, anti-insect activity. And we're also looking for ones that don't have any mammalian toxins in them. We're now going through the next step, which is inoculating them, and we've managed to inoculate several of these into rye and triticale. Rye and triticale we use because those particular species are a bit closer to the wild sort of species than say wheat or barley, which are a lot more domesticated. And at the moment, we've had a number that we've actually gotten through a full life cycle and transmitted the endophyte into the next generation of seed. And we're trying to understand at the moment some of the biological processes so we know when they switch on the mechanism to produce the alkaloids and when they switch it off. We need to understand some of those things. So we need to understand the bioactivity, and that's sending these out and running a whole heap of screens against our various insect pests. And last of all, we need to deal with safety. We need to understand that they're not producing any nasty alkaloids and that they're going to be safe for animals and humans alike. So it's a big program, and we're very fortunate to get a substantial slug of funding out of the New Zealand government as well in the form of a grant, which has enabled us to really speed the work up. We're about 4 years along now, and this come June, and we've just refunded the project for another 4 years, which will take it right out to commercial delivery. Our ryegrass that our cereal rye that we've produced with the endophyte in it has successfully gone through a number of generations, taking the endophyte with it, and we're looking now to go into barley as our next target. The last area I want to talk about today is microbial insecticides. So these are insecticides based on naturally occurring microbes from Australia. So the first area I want to talk about here is the issue of using things like organophosphates. So this is dimethylate in a pulse crop, and we're chasing mirids, but unfortunately the beneficial insects also get hammered when you use something like an OP, which is a broad spectrum insecticide. So you can see the first couple of days after that spray, the numbers of beneficials are absolutely hammered. What does it actually do then? Well, the mirids got knocked around, and that's great, but then the Heliothis, because of the natural predators disappeared, they peaked very quickly because there's nothing to take them out. So we've actually caused ourselves a bigger problem. So can we look at some of these fungus products as fungal biopesticides as very specific products to chase the aphids and thrips, but not affect the good guys, the beneficial insects? And so far, we've been going on this project for about 6 years now. We have a number of lead candidates that look as though they're going to work on a range of pests. We've already gone through and found a Metarhizium and a Bavaria. This is what it looks like when it was done on a cane grub, which is a product that's already released on the market from another company. But we know that they can work against sucking insects, and we have a range of good isolates that are working well on green vegetable bug, mirids, thrips and aphids. The next key will be, can we produce these in large amounts in an industrial process, and then can we make that into a cost-effective product and take it through a registration process? Last of all, I want to very quickly talk about a project that's only just started in the last couple of months, and that's looking at a granulovirus product for diamondback moth. We actually have a partnership with one of the companies to take this to market already. They want to play a part in the development role and have invested with us. We have a range of international and Australian isolates that we'll be going through, and we're looking at very specific, host-specific viruses. We aim to be in the market by about 2016-17, which is a pretty ambitious sort of an approach, but we're reasonably confident because viruses have been successful in agriculture, as many of you guys know. Products like Vivus Gold and Gemstar are based on viruses for Heliothis. We think a similar pathway will allow us to get to a product for diamondback moth. Before I finish today, I just wanted to quickly announce those of you that are interested in the Dgt Phosphorus Test. We've recently done a deal with APAL, and we're now working with a number of other labs to also be able to supply the DGT tests. So if you want to talk to APAL, they can do tests immediately. We're also hoping to release in the next couple of weeks a barley variety identification test. This will enable growers and advisors to be able to send a sample of barley seed to get an identification of the variety and the level of purity in that seed. We hope to have that out in a couple of months' time. Thanks very much.
Well, you explained that most of the insecticides have ultimately lost activity and some of them fairly quickly. And then you also explained that spiders have a myriad of different active compounds, and that's how they've avoided resistance evolving to the spider bite. Yep. And yet you seem to be going down a path where you're going to isolate and use a single compound. Have you given thought to actually using a blend of these?
We have, and one of the real benefits of working with this particular group is they're taking it to a molecular level in the understanding of the modes of action of these particular peptides. And that's something that we haven't done in previous work. Look, we know that if we can go in there with two peptides, one, for example, that might block a particular receptor, and the other one that might actually hold it open, and you use both of those, the chances of forming resistance to both modes of action is fairly low. So yeah, we're very, very cognizant that we need to get something that either reduces the chance of getting resistance to that particular one, or in some cases we may actually, by having this particular product in the market and available, open up the chance of using one of the old products as well to be able to use in combination.
Just changing tack a bit, Paul, I heard you speak at Jamestown Farmers Update on the snail control. Yep. And we've been waiting and waiting. What's going on?
Where are we up to? This coming season we'll be putting out at least 4 trials, is my understanding at this stage. Hopefully 3 snail trials and 1 slug trial. We now have more isolates. We had only 1 isolate last year when we went out and did some field trials. We now have 4 of them in commercial formulation. And if we get through this, then hopefully we'll make a move and start talking to growers about getting into the market.
Paul, we've got one up on the screen for you.
How far away is the soil water nitrogen probe? Okay, so we've developed the widget, as you saw. We've got them finalising the prototypes on those. The next step is taking it to the engineering group, and that's why I've gone to a group at South Australia, University of South Australia, who really do know a lot about things like soil physics and the ability of pushing a probe into the soil. So I'm hoping that within 2 years we'll actually have a prototype to take out and start testing. Where we go from there, we'll see how it gets picked up commercially.